Okay, let's unpack this. Imagine swimming through a coral reef, right? You see all the vibrant life, the fish, the coral structures. But what if I told you some of the most crucial things happening there are invisible?
Mm.
Like imagine light itself, not just sunlight, but light made by one creature guiding another one home.
That's exactly the kind of surprising partnership waiting in the sources you've shared. And these aren't just, quick encounters. We're talking deep, long-term associations, symbioses between different kinds of life.
Right.
Relationships so fundamental they really shape whole habitats and, well, the survival of species in them.
Yeah, exactly. So today's deep dive is all about these microbial symbioses, these
Huh.
really close, sometimes pretty astonishing partnerships involving microorganisms, whether they're teaming up with other microbes or plants or animals.
Mm.
And as we'll see, these interactions are often just absolutely critical to how life, functions on this planet.
And this stack of information you've got here gives us a fantastic window into this hidden world. It shows just how diverse and, frankly, inventive these partnerships can be.
Absolutely. So our mission today is really to pull out the core insights from all this material, understand the different ways these symbioses work, and just see how profoundly intertwined life is, especially, down at the microbial level.
Yeah.
Think of this as your shortcut, maybe, to getting informed on these amazing biological collaborations that underpin so much of the world around us.
Okay, so where should we start? That coral example you mentioned is pretty striking. How those vital algae find their coral hosts.
Yes. Let's start there.
Yeah.
It's especially relevant for young corals getting started or ones recovering from stress.
Right.
We're talking about the mutualism between stony corals, those are cnidarians, and these microscopic Symbiodinium algae. Now, unlike some partnerships where they're passed down, the coral often has to pick these algae up from the environment.
Especially when they're young larvae or juveniles, yeah. So you've got these tiny free-swimming algal cells in this huge ocean. How do they find the right coral?
This is where it gets really cool. The sources reveal the coral uses something amazing, green fluorescent protein, GFP.
GFP, like the stuff scientists use in labs all the time to tag proteins and make them glow.
The very same. I've always wondered what it did in nature.
Well, here the sources suggest a really beautiful function. The coral's GFP, it emits light. And researchers actually showed this light acts as a guide for the algae.
How'd they show that?
They did these experiments where they painted GFP onto little traps in the water, and the Symbiodinium cells, they were attracted right to the GFP traps. They just ignored the ones without it.
Wow. So the coral's basically putting out a little glowing welcome mat.
Essentially, yeah, a light beacon. And it works because Symbiodinium are naturally phototactic. They move towards light.
Okay.
But crucially, the wavelength of light from the coral's GFP, it perfectly matches the spectrum that's best for the algae's photosynthesis.
Ah, so it's not just any light. It's the right light for them. The coral is kind of hijacking their natural behavior.
Exactly. Leveraging their light-seeking instinct to draw them into the partnership. It's an incredibly specific signal.
Almost like a chemical attractant, but using light. That's amazing.
It is, and the sources really stress why understanding this is becoming more important, especially with coral bleaching.
Right, yeah. Bleaching is when they lose these algae, often because the water gets too warm. And if the corals can't get their partners back,
The whole animal is weakened, often dies. And that puts entire reef ecosystems, these huge marine habitats, at risk. So understanding how they reacquire symbionts is, well, it's key to their potential recovery.
it really shows the sophisticated signaling going on even between a big animal and a tiny microbe. Okay, so that's a macroanimal and a microbe. But these partnerships aren't just between big and small. Some really intricate setups happen just between microbes. The sources talk about microbial consortia.
Yeah, these are stable communities, multiple microbial species working together. Sometimes they're visually striking, too. A good example mentioned from freshwater is Chlorochromatium aggregatum.
Right, and that sounds like one organism, but it's actually a team, isn't it?
Exactly. The sources describe it as a collection. You've got non-modal green sulfur bacteria. Those are the phototrophs, capturing light arranged around a central bacterium that is modal but isn't photosynthetic.
So it's like a tiny, living, moving solar array. The outer bacteria make food with light.
They produce organic matter. And the central bacterium, it uses its flagella to move the whole group around, finding the best spot for light in the water.
And in return, it gets fed by the outer guys.
Precisely. It consumes the organic matter they produce. And the sources make it clear just how dependent they are. This link is often obligate, meaning they really struggle or can't survive without each other.
Obligate. So they're locked in.
Yeah, and the genetics back this up. Looking at the central bacterium's genome, it's lost a huge number of genes. Strong evidence it just can't make it on its own anymore. They rely on each other.
It's like a miniature ecosystem, self-contained, driven by this transfer of energy,
Yeah.
metabolites like alpha-ketoglutarate, maybe amino acids, moving from the photosynthesizer to the sort of engine in the middle.
And this points to a really core insight from the sources about these symbioses. They show incredible innovation in how energy and nutrients get passed between partners.
Right.
Which brings us to another type of microbial partnership discussed: direct interspecies electron transfer. Diet.
Diet. Sounds like something else entirely, but you mean microbial electricity.
Pretty much. The sources describe these methanotrophic consortia groups of microbes in anoxic marine sediments, so no oxygen. They work together to break down methane into CO2.
Which is a big deal environmentally.
Yeah, converting methane. That's a potent greenhouse gas. How do they manage that together? This is where diet is key. You have specific methane-eating archaea, called ANME, living really close to sulfate-reducing bacteria. Now, normally, microbes might exchange electrons using little molecules that diffuse between them, like hydrogen.
Okay.
But here, the ANME literally transfer electrons directly to the sulfate reducers.
Directly. How? What's the connection?
Get this. The sources describe electrically conductive appendages, essentially microbial nanowires that physically bridge the gap between the cells.
Nanowires, linking them up. That's kind of wild to picture. They're literally plugging into each other.
It really changes how we think microbes can interact, doesn't it? This direct electrical link, it's a fundamentally different way for species to cooperate on energy. And the sources suggest this diet thing might be way more common out there in microbial communities than we realized.
Wow. Okay, so from light signals and swapping chemicals to actual electrical wires, the ways microbes partner up are incredibly diverse. Let's shift gears now from microbes partnering with microbes to their crucial role with plants. The sources talk a lot about the famous nitrogen fixers, rhizobia and legumes.
Ah, yes, this is a huge one, absolutely vital, especially for agriculture. This partnership, where rhizobia bacteria live in legume root nodules, it accounts for something like a quarter of all the nitrogen fixed biologically on Earth each year.
A quarter? That's massive. It's why legumes like beans and peas are so good for the soil, right? They add nitrogen back.
Exactly. And the sources detail the really intricate process of how this partnership even forms. It starts with the rhizobia attaching to the plant's root hairs.
And that triggers something in the plant.
Yeah, a signal that makes the root hair curl around the bacteria. Then the bacteria induce the plant to form an infection thread, basically a tube made of plant cell wall material that guides the bacteria right into the root cells.
So the plant helps them get inside.
It does. And once inside, the bacteria multiply and transform into these swollen, kind of branched forms called bacteroids. They live inside specialized compartments within the plant cells called symbiosomes. That's the factory floor for nitrogen fixation.
Okay, but nitrogen fixation, that needs the enzyme nitrogenase. And I remember that's super sensitive to oxygen. How does the plant deal with that inside a living root?
Great question. The plant produces a special protein in the nodule called hemoglobin. You can guess from the name, it's very similar to the hemoglobin in our blood.
Ah, it binds oxygen.
Exactly. It acts like an oxygen buffer, tightly regulating the O2 levels, keeping them extremely low right where the nitrogenase is working. The plant creates and maintains the perfect, tiny, low oxygen bubble for the bacteria.
It's a custom-built habitat. And the whole process starts with very specific signals, right?
Yeah.
The sources mention bacterial nod genes.
Yes, absolutely crucial. These nod genes allow the bacteria to produce nod factors. These are specific molecules, lipid and oligosaccharides, and they're the primary signals. They're what trigger the root hair curling and tell the plant cells to start dividing to build the nodule structure.
It's a very specific molecular conversation to get things started.
Yeah.
The sources also mention some legumes can even form nodules on their stems, not just roots.
Right, like with azorhizobium. Now, moving from rhizobia, the sources draw this really fascinating link to another huge plant-fungus partnership, mycorrhizae.
Mycorrhizae, yeah. Those are the mutualisms between plant roots and fungi. The fungi basically extend the plant's root system way out into the soil with their hyphae.
Right, like a massive web.
And they bring back nutrients the plant struggles to get, like phosphorus and nitrogen. In return, the plant gives the fungus sugars from photosynthesis.
And there are different types depending on how close they get to the root cells. Ectomycorrhizae form a kind of sheath around the root tip and a network between the cells, the Hardick net, where they swap goods. But arbuscular mycorrhizae, or AM fungi, they go even further. They actually penetrate into the root cells.
Whoa, inside the cells.
Well, yes and no. As the sources point out, they don't break into the main part of the cell, the cytoplasm. They push the plant cell membrane inwards, forming these highly branched structures called arbuscules within the cell, but still technically outside the plant's main cellular machinery. There's a membrane barrier, creating a space called the apoplast where the nutrient exchange happens.
Ah, okay. Still very intimate, though.
Extremely. And here's the key insight the sources highlight. These AM fungi, they produce signaling molecules called myzoop factors. And guess what? They are also lipopide and oligosaccharides, structurally very, very similar to the nod factors from rhizobium.
Like the same type of signal molecule? That seems significant. An evolutionary connection?
That's what the sources strongly suggest. The idea is that the signaling pathway used for root nodule formation in legumes, which is, evolutionarily younger, maybe 60 million years old, probably evolved by borrowing from or adapting a much more ancient signaling system, one that was already being used by AM fungi in their partnership with plants, which goes back hundreds of millions of years.
So nature basically reused a successful communication system for a new type of partnership, like a common biological language.
Exactly. Pretty neat evolutionary story. The sources also briefly touch on a flip side, bacterial interactions that cause disease but have been harnessed, like Agrobacterium tumefaciens.
Crown gall disease, right?
Yeah, but it's ting plasmid, the thing that causes the disease by transferring bacterial DNA into the plant. That's become a cornerstone tool for plant genetic engineering, a natural gene transfer system we now use deliberately.
A reminder that understanding disease can give us powerful tools. Okay, let's zoom back in, relatively speaking, and explore the world of insect symbionts. Insects are everywhere, the most abundant animals, and these microbial partnerships are incredibly common and, well, key to their success.
Absolutely. These symbioses often provide stuff the insect can't get, otherwise essential nutrients, maybe protection. The sources talk a lot about heritable symbionts.
Meaning they're passed down.
Yeah, directly from parent to offspring, usually vertically through the eggs. And often, these are obligate relationships. The microbe absolutely needs the host. It can't survive freely.
And the sources break them down a bit. Primary symbionts are essential for the host even to reproduce. They often live in special organs, bacteriomes, made of special cells called bacteriocytes.
Right. And then secondary symbionts, they provide benefits, maybe help with diet or defense, but the host can reproduce without them.
Okay. And a really striking thing the sources mention about primary symbionts is their genomes.
Oh, yeah. Extreme genome reduction. Their DNA is tiny compared to free-living bacteria. They've basically thrown out genes for anything the host provides or does for them, makes them totally dependent.
Streamlined right down.
And their DNA often has this weird bias, lots of A's and T's, partly because they've also lost many of the enzymes needed for DNA repair.
Huh. And the nutritional help can be critical, right? Like aphids feeding on that sugary but nutrient-poor plant sap.
Classic example. They completely rely on their Buchnera symbionts to make essential amino acids that are missing from the sap diet. The aphid just can't make them.
Sometimes the sources say the pathway is even split, like the insect has genes for part of it, the microbe has genes for the other part.
Yeah, requiring both partners to finish the job. But the sources share an even crazier example. Get this. Mealybugs that host a bacterium, Morinella, living inside another bacterium, Tremblaya.
Wait, hold on. A bacterium inside another bacterium, like Russian dolls.
Basically. Layers of symbiosis. And these two nested bacterial partners cooperate to provide amino acids to the mealybug host. And that inner one, Tremblaya, has one of the smallest bacterial genomes ever found, like 0.14 MEP, just incredibly reduced.
That just underscores the extreme specialization you get in these obligate, passed down through generations partnerships.
It really does. It leads to a core idea from the sources. How a symbiont is transmitted really shapes its evolution. These vertically transmitted obligate ones, they tend towards tiny genomes and total reliance.
Makes sense.
And on the other hand, traits that symbionts provide can spread really fast through insect populations if they're transmitted well. The sources mention things like rickettsia and whiteflies, or Wolbachia.
Ah, Wolbachia, that does weird things to insect reproduction, doesn't it?
Yeah, like cytoplasmic incompatibility in mosquitoes.
Yeah.
It manipulates the host to ensure its own spread through the population. Very effective.
Okay, so beyond nutrition, the sources also talk about defensive symbionts, microbes as bodyguards.
Pretty much. They produce chemicals that protect the insect host from pathogens or parasites or even predators.
And the key detail the sources point out here is how they make those chemicals.
Right. Often, the genes for making these defensive compounds aren't part of the microbe's main chromosome. They're found on plasmids or other mobile genetic elements.
Meaning they can be swapped around more easily.
Exactly. It allows for horizontal gene transfer, sharing these defense genes between different symbionts, maybe even different species. It means the insect symbiont team can potentially adapt faster to new threats by acquiring ready-made chemical weapons.
That's a powerful way for the partnership to evolve. The sources give the example of the rove beetle.
Yeah, its Pseudomonas symbiont makes pterin, a really potent cytotoxic chemical defense, a weapon courtesy of its microbial pal.
Wow. Okay, one more insect example the sources dive into. Termite guts. Essential for eating wood, right?
Absolutely essential. Termites are masters at breaking down wood, but they can't do it alone. They rely almost completely on the microbes in their gut, especially in that big oxygen-free hindgut chamber.
And it's different for different termites.
Yeah. Lower termites, they have both anaerobic bacteria and these fascinating cellulolytic protists, single-celled eukaryotes that actually engulf and digest the wood particles.
Protists eating the wood inside the termite.
But higher termites, which are most species, they rely mainly on a super dense community of mostly anaerobic bacteria, including species that break down cellulose.
Okay, so the microbes do the heavy lifting, breaking down the tough plant fiber. What's in it for the termite?
The microbes ferment those wood building blocks, the polysaccharides, and produce volatile fatty acids, or VFAs, mostly acetate.
Like the VFAs in the rumen?
Very similar concept. The termite absorbs this acetate directly, and that's its main source of carbon and energy from its diet.
So it's eating microbial fermentation products.
That's the core of it. And the sources also highlight the role of spirochetes, those spiral-shaped bacteria in the termite gut. Many of them perform acetogenesis.
Meaning they make acetate.
Yeah, they take hydrogen and CO2, which are waste products from other microbes' fermentation, and they convert them into more acetate for the host using a specific pathway, the acetyl-CoA pathway.
So they're recycling waste products into fuel.
Exactly. And importantly, by consuming that hydrogen, they prevent it from building up. Hydrogen buildup can actually slow down the main fermentation reactions. So these acetogens and also methanogens, which consume hydrogen to make methane, they effectively keep the whole fermentation engine running smoothly, benefiting the termite.
They're like the cleanup crew that also provides extra energy.
You got it. And the sources also note some gut bacteria fix nitrogen, which is pretty scarce in a wood diet, helping the termite conserve that essential nutrient.
It's a whole tuned ecosystem in there.
Yeah.
And things like termite social behavior probably help pass these essential partners around, right?
Absolutely. Stable transmission is key.
Okay, from insects, let's broaden out again. Other invertebrates have amazing partnerships too. The sources bring us back to light. Bioluminescent bacteria.
Yeah, some marine bacteria naturally glow, and lots of marine animals team up with them. The Hawaiian bobtail squid, Euprimna scolops, is a really famous example the sources highlight. It's a major model system.
Why is it such a good model?
Well, it has these huge populations of just one bacterial species, a Vibrio fishery, living in a special light organ on its belly.
Okay, one species, that simplifies things. What's the partnership?
It's a beautiful mutualism. The light produced by the bacteria inside the squid mimics moonlight or starlight filtering down. This is called counterillumination.
So it helps the squid hide.
Exactly. It breaks up the squid's silhouette, making it much harder for predators looking up from below to spot it against the lighter surface water. Camouflaged by light.
And the bacteria, they get a safe house.
A very nice one. Nutrient-rich, protected inside the light organ, where they grow really fast. And interestingly, the squid actually vents or expels something like 90% of the bacteria out into the seawater every single morning.
Every morning. Why?
It seems to serve multiple purposes, but one key benefit for the bacteria is dispersal. It constantly seeds the surrounding water with a fishery, helping the bacterial population spread.
That daily seeding is quite a detail. So why is this squid such a research powerhouse?
Because you've got one host, one main bacterial partner, you can raise them easily in the lab, the symbiosis isn't strictly essential for the squid to survive in the lab, and crucially, the partnership starts fresh with each baby squid.
Ah, so you can watch it happen from the beginning. How do the baby squid get the bacteria?
It's horizontal transmission. They hatch bacteria-free and have to pick up a fishery from the seawater. The squid secretes mucus that attracts bacteria generally.
But how does it get the right one? The ocean's full of bacteria.
Great question. Chemotaxis is part of it. The squid releases specific sugars from pores on the light organ, and a fishery swims towards them. But Here's a really neat trick the sources mention. The squid also produces nitric oxide, NO.
Nitric oxide, isn't that usually like an immune chemical to kill bacteria?
It often is, but a fishery has evolved the ability to tolerate NO, even consume it. Most other marine bacteria can't handle it. So the NO acts like a specific filter. It deters or kills the wrong bacteria, giving a fishery a huge competitive advantage to colonize the light organ.
Wow, so the squid's own defense chemical helps select its partner. That's incredibly clever biology.
It really is. Now, from partnerships using light, let's go somewhere there's absolutely no light. The deep-sea hydrothermal vents and cold seeps.
Right, those ecosystems running entirely on chemical energy, not sunlight. And the big animals there, tube worms, giant clams, mussels, they're often totally dependent on internal microbes, yeah.
Completely dependent. They host chemosynthetic bacteria right inside their tissues. These bacteria are chemolithotrophs. They get energy by oxidizing inorganic chemicals like hydrogen sulfide or methane pouring out of the vents or seeps.
So the animal's body provides access to the chemical fuel.
Exactly. Take the giant tube worm, Riftia. It lives near black smokers, takes up toxic hydrogen sulfide from the vent fluid, and transports it safely via special hemoglobins in its blood to bacteria living in a big internal organ called the trophosome.
And the bacteria inside use that sulfide.
They oxidize it for energy. Then they use that energy to fix carbon dioxide into organic matter using pathways like the Calvin cycle, just like plants do with sunlight, but using chemical energy instead.
So the bacteria are basically feeding the worm from the inside out.
That's the essence of it. This organic matter is the worm's primary food source, allowing it to thrive down there. The sources suggest the worm probably digests some of the bacteria directly to get the nutrients.
A deep-sea internal farm. Now, thinking about those obligate insect symbionts with tiny genomes. What about these deep-sea guys? Are their genomes small too?
That's an interesting contrast, the sources point out. While many obligate symbionts, especially vertically transmitted insect ones, have drastically reduced genomes, the Riftia tubeworm symbiont actually has a relatively large genome.
Why the difference?
The thinking is it relates to transmission. Unlike the aphid symbiont passed down in eggs, Riftia worms acquire their symbionts horizontally from the environment when they're young. This means the bacterium needs to be able to survive for a time as a free-living organism in the harsh deep-sea environment before it finds a host.
Ah, so it needs more genes to cope with life outside the host.
Exactly. It needs a bigger genetic toolkit for that free-living stage. The genome size tells a story about its lifestyle.
Fascinating. Okay, one more invertebrate example where microbes are used essentially as weapons.
Right.
Entomopathogenic nematodes.
Right. These are certain families of tiny roundworms like Heterorhabditis and Steinernema that are specialized insect killers, obligate pathogens. And the key thing is they don't do the killing themselves. They use bacteria.
So the nematode is like the delivery vehicle and the bacteria are the weapon.
Precisely. The infective juvenile stage of the nematode carries its specific symbiotic bacteria Photorhabdis or Xenorhabdis inside a special pouch. It seeks out an insect host, burrows inside,
And then releases the bacteria.
releases them right into the insect's bloodstream, the hemolymph, and the bacteria just take over.
How so?
They multiply like crazy, produce powerful toxins that shut down the insect's immune system and kill the host, usually within a day or two. Then they release enzymes that liquefy the insect's insides, turning it into a nutrient soup for the nematodes to feed on, grow, and reproduce within the cadaver.
Grim, but effective. A coordinated attack.
And the sources mention that's usually a very specific nematode-bacteria pairing.
Very high specificity, yeah.
Yeah.
Suggests a long history of coevolution. It's a tightly integrated parasitic symbiosis. And the practical angle is biocontrol.
Exactly. These nematode bacterial complexes are studied and commercially produced as biological pesticides. You can spray the nematodes onto soil or plants. They find the pest insects, release their bacteria, and kill them. A targeted alternative to chemical sprays.
A potent tool straight from understanding a natural partnership.
Okay, we've covered a lot of ground corals, microbes, plants, insects, deep sea. Let's bring it closer to home now. The microbial world inside mammals, especially herbivores.
Right, this is maybe more familiar. Why do animals like cows or horses need microbes so badly?
It really boils down to their diet.
Ooh.
Plant material, especially the cellulose and hemicellulose that make up plant cell walls, is incredibly tough for mammals to digest. We just don't make the right enzymes, like celluloses, to break that stuff down efficiently.
So they absolutely need help.
They do. Herbivores have evolved these amazing modified beds with large oxygen-free fermentation chambers. These are designed specifically to house dense communities of microbes that can break down plant fiber via fermentation.
And these chambers hold on to the food for a long time.
Yeah, extended retention times gives the microbes enough time to work their magic. The sources outline the two main strategies for where this fermentation happens.
Foregut versus hindgut, right?
Exactly. Foregut fermentation, like in ruminants, cows, sheep, goats. The main fermentation vat, the rumen, is before the animal's true acidic stomach and small intestine.
So the microbes get first crack at the food, they break down the fiber, ferment the sugars.
And produce those volatile fatty acids, VFAs, mainly acetate, propionate, butyrate. The animal absorbs these VFAs directly through the rumen wall, and that's their major energy source, up to 70% sometimes.
Okay, what's the big advantage of doing it before the stomach?
The sources highlight a key benefit. After the microbes have done their work fermenting the fiber, the entire microbial population, which is incredibly dense, flows downstream into the acidic stomach.
And the stomach acid kills them.
Kills them dead. Then the animal digests the dead microbes in its small intestine. This makes the microbes themselves a huge source of high-quality protein and vitamins for the host animal.
So the animal eats the plant, the microbes eat the plant, and then the animal eats the microbes. It's incredibly efficient.
Very efficient nutrient capture. Now, the other strategy is hindgut fermentation. Think horses, rabbits, koalas. Here, the main fermentation happens after the small intestine in an enlarged cecum, or large intestine.
So the animal digests the easy stuff first, then the fiber goes to the microbes later.
Right. Fiber still gets fermented, VFAs are produced and absorbed, providing energy. But the downside is...
The microbes themselves, the protein source, mostly pass out in the feces.
Exactly. The bulk of that microbial protein is lost before the animal can digest and absorb it, which is why some hindgut fermenters, like rabbits, practice coprophagy.
Getting their own poop.
Well, specific soft fecal pellets, yeah. To recover those microbial nutrients they missed the first time around.
Makes sense, in a way. Let's zoom in on the rumen, since it's such a classic, complex example from the sources. What's that environment like?
It's huge. 100, maybe 150 liters in a cow. It's warm, strictly anaerobic, no oxygen. The pH is kept in a pretty narrow range, around 5.5 to 7. And food stays in there a long time, 20 to 50 hours, sometimes even more. Just perfect conditions for anaerobic microbes.
And the microbial community itself is massive.
Unbelievably dense. We're talking 10 to the power of 10 or 11 bacterial cells per gram of rumen contents, plus loads of archaea, anaerobic fungi, and protists. It's a whole ecosystem.
And the basic job is breaking down plant bits into sugars, then fermenting those sugars into the VFAs the cow absorbs.
That's the core process. They also produce gases, CO2 and methane, as byproducts, which the cow has to batch out.
Can't forget the burps. The sources mention key groups of microbes doing specific jobs.
Yeah, you've got specialists, bacteria that excel at breaking down cellulose, like Fibrobacter or Ruminococcus, others that are great with starch if the diet includes grain, then groups that ferment intermediate products like lactate or succinate, cleaning things up.
And the archaea, the methanogens like Methanobrevibacter, what's their critical role again?
Their main job is to consume hydrogen gas, H2, which is produced in large amounts by the fermenting bacteria. They combine H2 with CO2, or formate, to make methane, CH4.
Okay, methane is a waste product, even a greenhouse gas. But why is getting rid of the hydrogen so important?
Because hydrogen buildup actually inhibits many of the key fermentation reactions that produce the VFAs. It slows everything down. By constantly removing H2, the methanogens essentially keep the fermentation pathways running efficiently, allowing the bacteria to break down more fiber and produce more energy for the cow.
So they're like essential waste removers. They keep the whole engine running. Hydrogen sinks.
Exactly. They drive the whole process forward. And the anaerobic fungi and protists in there also play big roles, especially in physically breaking down tough plant fibers and contributing to fermentation.
It's such a complex, balanced system. But the sources also mention it can get thrown off balance easily, right? Like acidosis in cows.
Yeah, that's a classic example of disruption. It typically happens if you suddenly switch a cow from a high forage diet to a diet high in grain or concentrates.
Grain has lots of starch.
Right. And that readily available starch allows certain bacteria, especially one called Streptococcus bovis, to grow incredibly fast.
S. bovis isn't usually a major player.
Normally, it's there in low numbers, but with tons of starch, it blooms and ferments that starch very rapidly, producing large amounts of lactic acid.
Lactic acid. That's stronger than the normal VFAs.
Much stronger, so it causes a sharp drop in the rumen pH. That's acidosis.
And the low pH messes everything up.
Totally disrupts the normal microbial community, inhibits the fiber-digesting bacteria, can damage the rumen lining itself, and in severe cases, it can even kill the animal. The sources emphasize that you have to introduce grain diets gradually.
Why gradually?
It allows other starch-digesting bacteria, which are slower growing but produce less problematic acids, time to adapt and increase in numbers. They eventually outcompete the fast-growing lactic acid producers like S. bovis, maintaining a more stable environment.
It really shows how dependent the host animal is on keeping its microbial partners happy and balanced.
Absolutely. And on a positive note, the sources also mention protective microbes, like Synergist a bacterium that can detoxify mimicine.
What's a mimicine?
It's a toxic compound found in certain tropical legumes, like lucena, that ruminants might eat. Having S. in the rumen allows them to eat those plants safely.
So microbes aren't just digesting food, they're also running a detox service, a built-in defense system.
It's another layer to the partnership. So we've really taken a sweeping tour through your sources here. We've seen these incredible microbial symbioses everywhere, from guiding algae to corals with light to microbes wiring themselves together in mud, powering plants, giving insects superpowers, fueling life in the pitch-dark deep sea, acting as biocontrol agents, and of course, unlocking energy from grass in cows.
It's honestly mind-blowing seeing how these invisible organisms are just so fundamental to how the visible world works. And as the sources show, these aren't just, random microbes hanging out together. They're often incredibly specific, co-evolved partnerships providing absolutely essential things: food, defense, signals, energy conversion.
Absolutely. Life on Earth, even for us, for big plants and animals, it's fundamentally built on these hidden, vital microbial relationships.
And digging into these partnerships, pulling out these insights from the sources like we did, it really does give you a completely different lens for looking at the world. You think about a coral reef differently, knowing about the light signals. You think about farmland differently, knowing about nitrogen fixation and mycorrhizae. Even the insects in your backyard might have microbial bodyguards or digestive helpers.
It just underscores the incredible interconnectedness of life right down to the smallest scale.
Yeah. Which leaves us with a final thought, something for you, the listener, to mull over. If so many organisms we can see rely so completely on these unseen microbial partners for their basic existence, how many other complex systems out there in nature, maybe even inside us? How much relies on microbial communities that we're really only just beginning to get a handle on?
It's a vast hidden world, still full of secrets.